Okay, start when you're
Alrighty, we are ready to go. Alrighty, perfect. I'll go ahead and get started. Thank you everyone for coming. My name is Darrin Ambat, a current master's student at San Diego State University. I'm going to keep it short and sweet today. I'm going to start off by just giving you guys a brief background of who I am, some of the research I've done in the past, what I'm planning to do what I'm currently doing right now, and then how that all got changed when COVID started. But I'll wrap it up with more of a positive note of why this research, or why my research specifically, can still be important. If and when we get to that point, we'll move forward. So I started a long time ago, it felt like, doing research, terrestrial research with frogs in the middle of nowhere, tromping through rivers, working with the United States Geological Survey. And working on land really showed me that I don't like working on land. I actually ran into a bear, and there's just way too many bugs for me. So I decided, what can get me away from bugs and bears? I'm gonna go to the ocean. There's no bugs and bears in there. So when I started at a local junior college by my parents' house in Orange County at Orange Coast College, I really tried to focus a majority of my classes on marine science classes. And from there, I just started loving and falling in love with everything I could learn about marine science and marine organisms specifically. So I looked into schools, and I came across CSU Monterey Bay up north, kind of central California, and I just fell in love with the marine science program. The people there, the classes, they just had a lot to offer. So I just started expanding this career. I actually got into undergraduate research at the National Oceanic and Atmospheric Administration up in Santa Cruz. They have a nice Southwest Fisheries Service up there. So as an undergrad, I had an opportunity to look at how ocean acidification and hypoxia affects both the blue and brown rockfishes. And I specifically worked in a lab, so a lot of lab work maintaining fish, aquaria, and just kind of learning as much as I could as an undergrad. And I even got to do my own personal individual research project. So I looked at how blue and brown rockfish larvae, in this kind of middle picture you see right there, how their larvae are affected by hypoxic conditions, so low amounts of dissolved oxygen. So I found out that, yeah, low oxygen does affect these larvae. It could stunt their growth. They could have anything from eye deformities, but it really did affect these fish. So how are hypoxic zones that are caused by human disturbances such as runoff, how is that affecting fish? So I got really interested in, okay, human impacts on fish. This is exactly what I want to do. It's working with fish. I'm good to go. But this was a lot of microscope work, and undergrad was awesome, but it was just too much time in the lab for me. I wanted to get out, get in the sun. And at this same time, I started getting into diving. So I was like, okay, what can I do that's good diving, but still working with fish and human impacts? So I came across and got this really cool opportunity to actually work out of the Wrigley Institute a few summers ago now. And it was a really dive intensive program. So what we did is we studied these two sex changing species of gobies. I was seeing these top pictures right there. We have the black-eyed goby, and we have this cute little blue-banded goby. So this was a really dive heavy project. As you can see, it looks like a lot of us are tired laying on the dock right there between dives. We did anywhere from on average, about 3 dives every day, all day for the entire summer. So just think about that we did about 175 dives over the course of 3 months. So we had like very few days off, but it was awesome. It was really immersive. It gave me a lot of background working with graduate students, PIs, professors, all working together to accomplish this big overall project, and how the little bits can branch off of what graduate students are doing as little offshoots to the main project. So it was a lot of diving, a little bit of lab work still, but it was definitely the flip of what I did before, and all in the lab. So I have the lab experience now, and that built up that field component. But honestly, as much as I love diving, it got a little tiring doing 3 dives a day, every day, all day. So I decided when I entered grad school that my project would hopefully be a 50-50 mix, maybe just a little less diving to keep, my volunteers sane. So I fortunately got into the master's program at San Diego State in Dr. Matt Edwards' Kelp Ecology Lab. We're also known as the Beer Pigs or Benthic Ecology Experimental Research Ecology in General Lab. That's a mouthful, so we just go by the Beer Pigs. We also have a branch up in Moss Landing, which is super cool to collaborate with them. But basically what I'm doing is I'm carrying on my research that I've done, my background with fish, so the blue-banded goby specifically this time, but I'm using these benthic chambers as you see in this picture on the right. So I'm setting up these artificial reefs, and I'm putting basically a bunch of rocks and a pH sensor and a dissolved oxygen sensor in this benthic chamber. And I'm putting a bunch of fish in there. So I'm putting blue-banded gobies in there with these little PVC tubes that they actually lay their eggs in. We call these, not a scientific term at all, but we call them tunnels of love, or TOLs for short. They're like little goby hotels, so they can lay their eggs there. And then with these benthic chambers, they isolate this completely fixed volume of water inside this chamber. So what I'm able to do is to study ocean acidification, I'm able to acidify the water within those chambers. So we can have different acidified treatments, and we can have also control treatments and have little kind of mesocosms on the benthic floor of our study site. And what you can do is you can pull out, see these little goby hotels in these pictures on the left. The gobies lay their eggs on this little sheet of paper rolled up in the tube, and we're able to pull those out and photograph them while underwater, and we're able to count the eggs. We can see how many are fertilized, we can measure how big they are with image analysis programs, and we can really track how the pH and the dissolved oxygen is changing within that chamber, since it is that fixed volume of water. And this is very beneficial because my research will incorporate the effects of ocean acidification that we've seen in the lab and from previous papers, but we're taking it into the field using this benthic chamber technology. So it's kind of a novel idea. It's like, how do we study OA in the field? Because this really improves the design for field deployed chambers that really hasn't been done up to this point, especially with fish. And these chambers are awesome because they incorporate visual cues such as from predators or that of light, which can both be really important for fish reproduction. And it also incorporates the natural and chemical and physical characteristics that we see in a wild natural habitat that's really hard to replicate in the lab. Another example of why this study is going to be beneficial is because most studies use a static, or they use like one steady value throughout the whole study for their pH. So they might use like an ambient kind of control pH, and then they'll use an acidified, and they'll just keep it at that same level throughout the entire study. However, we know that pH varies on both time and spatial scales. So anywhere from like day and night, just due to some processes such as photosynthesis and respiration. So that pH is always changing in a natural habitat. So why wouldn't our study want to mimic that? Well, that's what my study is going to do. It's going to use this it's going to allow that pH to fluctuate like we see with this red line. It's going to fluctuate compared to the ambient conditions, but it's going to remain in that acidified condition. And we have already been working on it at Wrigley before some of the closures, so I was able to do some preliminary research using this chamber technology. The aim of this preliminary research was to just, let's see if these chambers work. Let's see if we can acidify the chamber. So thankfully we did. We successfully acidified those chambers by about the 0.2 pH units that we see for future pH projections of the ocean. And we're able to do that. It wasn't perfect, but it's getting there. Hey, we're starting, we got some good data. And as you can see in these green pictures, we use this dye to dye the water that's being injected into those chambers. And you can see, wow, that chamber really does isolate that fixed volume of water. Maybe a little bit of leaking in that picture labeled B, but hey, we can fix that with improving our methods down the line. So good job. We got some good data this past trip. We were on Catalina out of Wrigley. But then as I'm ready to go, everything kind of starts to go sideways. So COVID started and restrictions and things started to close. So now what do I do? I had everything planned to be on Catalina, but let's not let that bad news get to us. So we're going to transition everything into a lab. It could be a quicker way to set up the same experiment. So what I'm doing is I'm actually staying in San Diego where I live and where I go to school, out of the marine lab there. It's called the Coastal and Marine Institute Laboratories, or CMIL for short. And I was able to set up these laboratory mesocosms to pretty much just have the same thing that I was going to do in the field, but this time I'm going to do it in the lab. I'm going to have control pH tanks, and I'm going to have acidified pH tanks, and have those TOLs, those goby hotels in there to see how ocean acidification is affecting this blue-banded goby reproduction. So not too bad. We still get to do it in the field or in the lab, and hopefully once everything opens up, the plan is to take all of our stuff, head out to Wrigley, and just start cranking away at field work. So not too bad, not too shabby. Pushed us a little back just because you have to get permits and redo kind of everything for a different location, but it's okay. We figured it out. We're adapting. It's definitely been a learning experience for sure. So not too bad. And the significance of studying ocean acidification in a field setting instead of just the laboratory, it really gives us a more increased ecological realism. So looking at how natural chemical fluctuations, such as how pH varies, it could be predator cues, light cues, anything that you find in the natural habitat, you can't really mimic in your laboratory mesocosms. So once we get in the field, we'll be able to do that. Blue-banded gobies are a good model species for other ecologically and economically important fish species, such as the California sheephead, as well as other sex-changing fish. And really this, my study is going to expand our understanding of how to better understand or better study OA and the effects for nearshore organisms that experience this high amount of variability in seawater conditions. So that fluctuating pH that we see, especially in coastal conditions. And it's really going to give us this improved methodology of how to study OA effects in the field. So we're able to take laboratory mesocosm studies that we've seen in the past and really shift that to the field to make it a more natural habitat. So all in all, this study is going to help us understand ocean acidification in a more broad, natural setting. So short and sweet, and if you guys have any questions, I am available. I put my email at the bottom left just in case anybody would like to reach out and contact me with any questions. If you can't think of anything now, I'm always open to chat. My research past, how I've gotten to where I've gotten, some of the research experience I've had, or even just anything from, hey, these fish are cool. Can you tell me a little bit more about them? And maybe ocean acidification, we can always nerd out about that kind of stuff. So I'd like to thank everyone for coming, and I hope you guys enjoyed it and at least learn a little something.